Hey, my name is Matt Robertson, and I'm going to discuss a review on color vision deficiency involving the C203R mutation, as well as future curative options for those that suffer from color deficiency. I should start by saying that I have a form of color deficiency. I mix up red, green, blue, and purple all the time, and everyone's always asking me what color things are. My experiences with having this disorder caused me to want to dive into why it actually occurs. First, let's start with a brief background. Color vision deficiencies are present in about 8% of men and 0.4% of women. The mutations that cause color vision deficiency are located due to changes on the OPSIN1, LW, MW, and SW genes. These genes produce proteins that are essential for normal color vision, and they're located in the retina. The LW, MW, and SW in the genes refers to the wavelength of light that the cones are associated with. For example, LW is associated with long wavelengths of light, while MW and SW are associated with medium and short wavelengths respectively. The mutations that occur in these OPSIN1, LW, MW, and SW genes are located on the X chromosome, and color vision deficiency caused by these mutations serves as a classic model for X-linked recessive inheritance. Now let's discuss the actual C203R mutation. This mutation is the most common one that causes red-green color deficiency. It's a missense point mutation where a single nucleotide change results in the coding of a wrong amino acid. In this particular case, a substitution of cytosine for thymine in nucleotide position 1101 results in the substitution of arginine instead of cysteine in amino acid position 203. This mutation occurs in the OPSIN1 MW gene located at the XQ28 position on the X chromosome. This mutation disrupts proper folding and stability of the opsin proteins coded for by the OPSIN1 MW gene. The tertiary structure is broken due to a disrupted disulfide bond that doesn't form because of the replacement of cysteine with arginine. This misfolding keeps the opsin pigment trapped inside the endoplasmic reticulum, which does not allow the pigment to be adequately transported to the cones. You can think of the mutation as something like a building that's collapsed and trapped people inside. Without help, they're unable to escape. A couple of experiments have observed results caused by the C203R mutation. In order to observe differences that the C203R mutation causes, cone density was compared from two individuals with the mutation compared to an individual without the mutation. Cone mosaic images at 4 different locations were taken and observed on a field of 90 by 90 micrometers. An observed difference in cone order and density could clearly be seen with the mutant cones being scarcer and less grid-like. It was only the MW cones that were affected. Another experiment observed estimates of normal retinal and outer nuclear thickness. These estimates were obtained using the Stratus OTC and retinal nerve fiber layer scanning. This compared the thicknesses between the two mutant subjects and 93 controls. The results for the total retinal thickness showed no significant retinal thinning. However, ONL thickness was outside of 2 standard deviations of the mean for the controls. Currently, there's no treatment to cure the many forms of color vision deficiency in humans. Fortunately, a new breakthrough for reversing color vision deficiency is on the horizon. This is thanks to the work of Dr. J. and Marie Nitz, who are professors of ophthalmology at the University of Washington. They're working on a novel treatment for color vision deficiency that has already proved to be effective in monkey trials. This treatment involves an injection of an adeno-associated virus. This allows for the addition of normal copies of a correct desired gene into the actual cones of the retina. Human testing will start in 2017. Thank you for listening to this mini review about color vision deficiency, and let's hope for exciting results from the experiments that are going to be happening soon in the future as scientists are working to reverse this condition. Thank you.